ICO, or IcoSeq, is the first adaptation of high-throughput RNA sequencing to microbes.
This method assesses microbial function at a genome-wide scale. For engineered microbes, this method can elucidate how changes to the microbial genome can perturb its functions.
Obtain yeast from a suspension culture and count the cells using a hemocytometer. Resuspend the cells in PBS at a concentration of about 750,000 cells per milliliter. Next, mix ultra-low melting point agarose in PBS and heat the mixture at 90 degrees Celsius until the agarose melts. Load the agarose mixture into a syringe with an attached 0.22 micron filter. Place a syringe pump in front of a space heater set to 80 degrees Celsius and place the syringe in the pump. Fill a second syringe with the yeast suspension and fill a third syringe with fluorinated oil with 2% ionic fluorosurfactant. Load both syringes into the syringe pumps. Connect the tubing from the syringes to device A. Place a 15 milliliter conical tube in an ice bucket and guide the outlet tubing into the conical tube. Set the flow rate for each syringe and collect approximately 1 milliliter of emulsion in the 15 milliliter conical tube. After waiting 5 minutes for the agarose in the tube to set, add an equal volume of 20% perfluorooctanol in fluorinated oil to the emulsion. Mix the emulsion and the perfluorooctanol by inverting the conical tube a few times. Centrifuge the broken emulsion at 2,000 times.